Today is going to be so sweet, so fat, and we're going to get totally pumped as we talk about the catabolism of carbohydrates, triacylglycerols, and proteins. When we left off last time, you all had built an epic electron transport chain, or I should say many electron transport chains across the inner membrane of the mitochondrion, the huge mitochondrion that we had placed into the stem foyer, and very reminiscent of the crowding and urban type environment that we see across the cristae, the infoldings of the inner mitochondrial membrane. We built different complexes. We all acted out the parts of different complexes within the electron transport chain. This image shows a very static, much less interesting version of the electron transport chain, helping us recall the roles that complex 1, NADH dehydrogenase, complex 2, the Wonder Woman enzyme, complex 3, cytochrome BC1 complex, and complex 4, cytochrome C oxidase, all played in that symphonic process of shuttling electrons and taking the energy from the shuttling of electrons to pump protons. And recall that we were able to note the number of protons that were pumped from those electrons that were shuttled. So 4 protons being pumped at complex 1, 4 at complex 3, and 2 at our terminal oxidoreductase, cytochrome C oxidase, adding up to a total of 10 protons pumped per NADH, or if we're tracking the process by which succinate is oxidized, creating FADH2 within complex 2, then we know that only 6 protons are pumped there. So we then followed those protons and their epic journey as they then ended up entering in at the ATP synthase machine, which turns like a turbine, converting one form of energy into another and allowing the synthesis of 3 ATP for every 10 protons. So we calculated those epically important conversion factors. Remember, 10 protons pumped per every NADH. And of course, we can convert that to the number of ATPs per NADH, noting 3 ATPs per every NADH. You all went ahead and calculated the conversion factor for the FADH2 as well, and managed to run through the full-on calculation for the number of ATPs that were net from the degradation of a glucose molecule from a single glucose molecule passing through the entirety of chemoheterotrophic metabolism. We begin the journey that a glucose molecule follows when it enters into catabolism at glycolysis, or the Ebden-Meyerhof pathway. And remember, in that Ebden-Meyerhof pathway, 2 ATP are net via substrate level phosphorylation, and of course 2 NADHs are net in that glycolytic or Ebden-Meyerhof pathway. Now let's bring over a picture. Now hopefully your posters look a lot nicer than mine. Mine gets a little chicken scratchy when I write it here on the computer, but we can take a quick look at the Ebden-Meyerhof pathway within the cytosol of a yeast cell and note that yield. So recognizing that that at the step 1 and step 3, we have to invest an ATP in order to then later get ATP out, and then finally when 1,3-BPG is formed, we yield 2 ATP, and in the very last step, remember, when phosphoenolpyruvate is converted to pyruvate, that's when we hit the pay dirt and actually net those 2 ATP via substrate level phosphorylation. So that's our generation of ATP there, but don't forget the NADH. I mean, NADH, it's formidable. It's worth quite a lot of energy if it can get its electrons to the electron transport chain. Of course, that requires that the electron transport chain is running. It also requires that the NADH can get to the electron transport chain. So let's take a moment to look at what the NADH has to go through in order to get to the electron transport chain within a eukaryotic yeast cell. It's going to have to get its its electrons across the inner mitochondrial membrane and drop them off at complex 1. But the trouble is that NADH does not have a transporter across that membrane, and so it cannot easily get its electrons to complex 1. It actually has to go through quite a lot of hoopla in order to get its electrons into the matrix of the mitochondria. It has to transfer them to another molecule, and then that molecule has to get in, and then that molecule has to transfer them back to an NAD+. So there is a lot that has to happen for that to take place. So the NADHs that are made within the cytosol do not just simply cross that mitochondrial inner membrane. And so we have to take note that cytosolic NADH is worth less. So let's go back to our calculation and note that generally we make the assumption that the cytosolic NADH is worth about 1 less ATP. So rather than that 3 ATP per NADH, we say there's an investment of about 1. It really depends upon what transport system is used, but we have to make an assumption that that NADH is not worth the full total. Now, as we move into the transition step, now that NADH is of course generated within the matrix of the mitochondrion, because remember there is a transporter, that pyruvate translocase, that gets the pyruvate into the mitochondrial matrix. So when the transition step takes place, the NADH generated is inside of the matrix, so it doesn't need that top secret clearance. It's already in the matrix. So again, we can peek at that on our picture, noting that the 2 pyruvates are within the mitochondrial matrix when the transition step takes place, when those 2 NADHs are yielded. So it's really quite straightforward for them to bring their electrons over to complex 1 or NADH dehydrogenase. Now the same could be said of those that are generated within the TCA cycle. These NADHs can also deliver those electrons straight to complex 1. There's no need for them to go through any finagling in order to get there. They just deliver them directly. So in that case, we know that those electrons are worthy of the full 3 ATP. So as we go back to our calculation, we can look at the yields adding up. You know, the 6 NADHs, 2 turns of the TCA, 3 NADHs per turn, 3 ATPs per NADHs, 2 FADH2s per turn, or 1 FADH2 per turn, 2 turns, 2 FADH2s in total, 2 ATPs per FADH2, and then of course in step 5 we have that ATP netted via substrate level phosphorylation. So hopefully everybody on that calculation added up to get 36 ATPs, and per glucose in this particular case where we were seeing the cytosolic NADHs worth less. So that being said, let's talk a little bit about the fact, in fact, some of you asked me just the other day about this. Glucose is not the only fuel out there. You know, there are many other fuels like fructose. I mean, fructose is the natural fruit sugar, or maybe you've heard of galactose or mannose. These are also important sugars, important monosaccharides, and they too can enter into the Ebden-Meyerhof pathway. Notice that they have to go through a slightly different process in order to add into the intermediates that we already know and love. Galactose, there are several steps that allow it to get converted to glucose 6-phosphate. Fructose and mannose, they can get converted into fructose 6-phosphate. This is why we call this the point of no return. Step 3 in the Ebden-Meyerhof pathway, remember we've called that the first committed step because it is the step that occurs after all of the sugars really have technically entered into the pathway. There is some caveat there because high fructose corn syrup does some other weird things and sometimes bypasses this heavily regulated phosphofructokinase catalyzed step. So remember that one is that epic enzyme PFK1. Yeah. So we also should take a look at the fact that not all sugars, of course, hopefully your diet is not filled with only monosaccharides, and instead you also have a great deal of complex carbohydrates in your diet. And so those complex carbohydrates likewise can add into the catabolic pathways. This requires the synthesis of enzymes that will cleave the glycosidic linkages within the polysaccharides. Alpha amylase cleaves a 1,4-glycosidic linkage. Oligo-1,6-glucosidase, you won't be surprised to know that it cleaves the 1,6-glycosidic or glucosidic linkage. You'll note that this looks like a complex polysaccharide with branches, and so this may be a starch that we commonly take in our diet called amylopectin. So not only are starches catabolized by us, but of course they're also catabolized by bacteria. So one of the ways in which we very commonly differentiate bacteria is by looking for their ability to digest starches. We have a particular biochemical test that we will do in lab where we put our bacteria onto one side of a plate that's filled with starch, and then we let our bacteria grow overnight. That means that the bacteria that can secrete alpha amylase and oligo-1,6-glucosidase will secrete those enzymes into the space around the cell and digest those into their smaller subunits and take them into the cell. So then if the next day after growing it, if we put iodine onto the plate, the iodine binds to the starch, and if the starch has been used up in the region around the bacteria, there's nothing to bind to. So you get a halo around the growth. This is a very cool positive starch hydrolysis test showing us that this bacterium, maybe something like Bacillus subtilis, it's positive for starch catabolism, has secreted those enzymes and is digesting the starch. Now, of course, the only polysaccharides out there are not these, what we think of as digestible starches, but of course there's also cellulose. And many bacteria would think we're crazy not calling cellulose digestible. And one particular area in which you'll find those bacteria that love to digest cellulose are in the rumen of cattle. This is one of my prior students, Lisa, and she is clearly feeding her cattle what they love, hay, which is packed full of cellulose. Now the cellulose reaches the rumen within these cattle, and the bacteria within the rumen will digest the cellulose into a disaccharide called cellobiose and then into free glucose. And then fermentative bacteria take up that glucose. They break down into mixed acids like acetic acid and butyric acid and propionic acid. And then that actually can serve as a source of energy for the cattle. We also see some really cool anaerobic bacteria within the rumen. We'll talk more about those later. But overarchingly, there are so many bacteria within the gut of cattle. There's we sometimes refer to them as being a 40-gallon fermentation vat, and really their rumen is just this immense fermentative reservoir. Now, of course, we also have bacteria that see things that we would never dream of as being digestible as their energy sources. For example, some bacteria digest TNT, some digest oil, we've talked about that before, and some digest things like pesticides. And all of those have to somehow get into those typical catabolic pathways that we've talked about. We call these compounds recalcitrant because they're not digestible by most life, but there are certainly bacteria out there that can do that digestion. And of course, as one of our poster groups will tell us, there are also fungi that are capable of doing that as well. Don't forget protein. Remember that protein is not the preferred fuel for life because it's toxic, quite simply. Every time an amino acid, so if this is our polypeptide chain traveling amino to carboxyl, we know that when a peptide bond is broken, it frees an amino acid. And we know that our amino acid has its amino group, carboxyl group, and its R group. I'll leave you to recognize really quickly what that R group is. But while you're thinking about that, recognize that in order for amino acids to get degraded, they have to lose their amino group. That forms ammonia, which is toxic and has to be cleared. For us, we clear it as urea. Not all organisms do. But that's one way of getting rid of it. So deamination has to take place. And you might get a hint as to, if you're not remembering what this amino acid is, which I bet most of you are because you're seeing that negative charge and you're like, this is the acidic one, glutamate. So glutamate can get deaminated to form alpha-ketoglutarate. We know that's an intermediate of the TCA cycle. And so when the deamination occurs, this amino acid directly fuels the TCA cycle. We call that an anaplerotic reaction because it fills up the TCA cycle and gets the TCA cycle going faster. So this anaplerotic reaction can take place. We can recognize that not all amino acids would enter into the TCA cycle. For example, if we were instead looking at, and I'll try to just jot down the amino acid alanine. So remember that it has the fairly simple R group being just a methyl group. So this is just a CH3 or a methyl. And then, of course, it's typical carboxyl and amino groups. So if this gets deaminated, loses that toxic ammonia, and then this, of course, changes this carbon. This is Gabby. You all haven't maybe met. I can't remember if you've met Gabby yet, but this will convert this directly into pyruvate. And of course, remember that's the ending molecule of the Ebden-Meyerhof pathway. So not all proteins, or not all amino acids, when they're degraded, enter into the TCA cycles, just some of them. Now, today, our challenge in class, and what we're going to spend our time working on throughout class today, is lipid catabolism. So as we take a look at a fat, recognizing that in order for a lipid to be digested by a bacterium, it would have to secrete lipase, and lipase would break the ester linkages in the lipid, leaving the long fatty acyl side chains and the glycerol backbone. We're not going to follow the catabolism of the glycerol backbone because oftentimes in a cell that's degrading fat as an energy source, the glycerol backbone is actually being used as a precursor metabolite in the anabolism of glucose, actually being used to build glucose. So we're not going to calculate the energy that would come from that glycerol backbone. But what we are going to do is to calculate the energy that will come from the fatty acyl side chains of this fat. So we have to then think about where does it enter if we're calculating that yield, and recognize that these fatty acyl side chains are degraded 2 carbons at a time. And where do we see a 2-carbon molecule in catabolism? Yeah, you got it, acetyl-CoA, or the top of the TCA cycle. So as this is degraded 2 carbons at a time, acetyl-CoA is formed. Now, there's a whole pathway involved in degrading that fatty acid side chain called beta oxidation, but we won't go into the depths of beta oxidation. Let's just see if we can calculate how much energy would come from the TCA cycle from all of these acetyl groups. Now, one thing that we have to make sure we do is to count the number of carbons correctly. And sometimes that can be a challenge if you're not sure where those carbons are. So this is our first carbon here. So if this is carbons 1 and 2, right? And then we have 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and 14. So 14 carbons. How many acetyl groups does that mean we have? And then how much energy are we going to get from this fat? So this is the challenge I'm going to leave you to work through during class and to calculate.